Good morning and welcome to Medicine Grand Rounds. Dr. Roman is not here today and he has asked me to introduce our speaker, which is a pleasure for me to do. It's Dr. Steve Winters. Of course, he has been Chief of Endocrinology here for 13 years, so I think everybody knows him well. I'll just say a few words about his academic background. He's a graduate of the State University of New York in Syracuse. Steve did his residency in medicine at the University of Rochester in New York, and then went to the NIH to do a research fellowship in endocrinology at the National Institute of Child Health and Human Development, and he left about the same year when I arrived there in 1978. He was recruited by the University of Pittsburgh as assistant professor, rose through the ranks to the rank of full professor, and in 1999 Dr. Redinger recruited him here as chief of endocrinology at the University of Louisville. Steve has received numerous honors and distinctions in his career, chief among which is the membership in the American Society of Clinical Investigation, one of the two most prestigious societies to which American physicians can belong. He is a reviewer for virtually every endocrinology journal. He's on the editorial board of Endocrinology and other journals, has been a reviewer for the NIH in many study sections and special emphasis panels, as well as a recipient of numerous NIH grants. He has published over 100 papers, original papers, in endocrinology journals and more than 50 reviews and chapters and editorials. And his research and contributions have focused mainly on male hypogonadism and the expression and control of GSH receptors and the etiology of male infertility. So he has been a major component of our department for many years, has made major contributions to research, training, and clinical activities, and we are very happy to listen to his presentation today on the endocrinology of hereditary hemochromatosis.
Thanks, Roberto. I hope my mother was listening. Okay. So I generally, maybe we can turn down the light in the front. There we go. So I generally like to begin my grand rounds with a case, and this is a patient who is seen by our consult service just this fall. A 42-year-old woman who was in Jewish hospital, and we saw her because of diabetes, and she had cirrhosis and non-alcoholic steatohepatitis. And as a teenager, she was overweight with irregular menses and acne, and she married at age 20 but had difficulty conceiving. Ovulation induction, presumably with clomiphene, was effective, but she had 2 macrosomic babies in pregnancies that were complicated by excessive weight gain and preeclampsia. At age 34, she developed diabetes and was treated with oral hypoglycemic drugs. Thrombocytopenia was found at age 38, and she was diagnosed with cirrhosis. Her blood glucose rose, and she was instead treated with insulin, and the following year she developed ascites. And the serum iron over TIBC was 196 micrograms over 253 micrograms at 77%, but the serum ferritin level was within the normal range of 276 micrograms per liter. And she developed amenorrhea. And by 2012, the serum iron rose further, and the transferrin saturation tended to decline. And now the ferritin was over 1,000 micrograms per liter. Genetic testing for the HFE hemochromatosis gene was performed, and it revealed heterozygosity only for the H63 mutation, while the C282 was wild type. And on 10/26, she had an orthotopic liver transplant, and her liver was diffusely cirrhotic with a hepatic iron index of 1.1 microgram per gram, which is adjusted for her age. And the normal value is less than 1, and values of 1 to 1.9 are only mild elevation and are nonspecific for iron accumulation. And what struck our consult service when they saw her for the diabetes and prompted them to recognize the clinical findings and put them together and order the genotype was this the brown-gray pigmentation, which was found throughout her body, and we took a picture of her feet. This is a very, very vibrant area. Hemochromatosis, if one does a Medline search, there were over 700 publications in 2012. And this case was just generated so much discussion in our group that I thought I would share with you from an endocrinologist perspective some of the recent advances in the molecular mechanisms for systemic iron regulation, to understand how iron overload occurs in hereditary hemochromatosis, and then to discuss clinical findings in patients with this disease, with special reference to dysfunction of the endocrine glands, most notably the gonadotropes. So iron is the second most abundant metal on Earth, falling closely behind aluminum. And all organisms, both unicellular and above, are dependent on iron for survival, because iron is a cofactor for proteins and numerous enzymes. The heme proteins, those that are bound with porphyrins, which include, of course, hemoglobin, catalases and peroxidases, cytochromes, and a variety of non-heme proteins involved in DNA synthesis, cell proliferation and differentiation. The cytochrome P450 is involved in drug metabolism, the steroidogenesis hormones, and a variety of other hormones involved in gene regulation. This is a picture of the iron cycle, and there are four pertinent cell types. The duodenal enterocyte, where iron is absorbed from the GI tract. The erythroid precursor in the bone marrow, where iron is incorporated into hemoglobin. The reticular endothelial macrophage, where those red cells are engulfed and digested, and the liver, hepatocyte, which is the main storage organ for iron. And about 25 milligrams of iron is reutilized each day, because the erythroid precursor cells, the red cells, then undergo senescence, and they're engulfed by macrophages. And the macrophages then store the iron together with a macromolecular protein known as ferritin. And then they release that iron, and it becomes bound to a transport protein, which is a high affinity binding protein known as transferrin. The transferrin is normally about 25% saturated, so there's a substantial amount of available transferrin binding sites. And transferrin tends to buffer the circulating iron and keeps it in this macromolecular form and allows it to enter into cells in a physiological manner, whereas iron, which is unbound, tends to damage cells. About 25 milligrams a day is reutilized, and in contrast then, about 1 or 2 milligrams a day of iron are lost, either by the skin or intestine, sloughing of cells or bleeding, and in women by menstruation. And this is then compensated for by iron absorption from the GI tract. The loss of iron, of course, is not regulated. Bleeding is not a regulated process, and thus iron metabolism is maintained in a neutral state by the exquisite control of the amount of iron in the diet, which is absorbed by the duodenal enterocyte. And you can see that the relative dietary allowance for iron is twice as high in women as in men, of course, to compensate for menstrual bleeding. And it turns out we probably get a lot more iron than we need in our diet. A large hamburger is about 8 milligrams. So this shows a close-up of a duodenal enterocyte. The iron in the GI tract is converted from the ferric form to the ferrous form and is absorbed into the duodenal enterocyte by a transporter protein known as DMT1. And once inside this cell, as in the macrophage, the iron can be partitioned and stored in part in this macromolecular complex known as ferritin. And in addition, some of the iron then gets reabsorbed into the circulation through a transporter known as ferroportin. And ferroportin then delivers the iron into the plasma, where it's converted to the ferric form by ferric oxidase, and it again is bound to transferrin. Now, the key regulator of iron absorption is this protein, hepcidin. It was identified in only 2000 as LEAP, liver endogenous antimicrobial protein number 1, because when it was purified from plasma and urine, it had antimicrobial activity, and it was structurally homologous to other antimicrobial proteins, and it was called LEAP in this paper in Dept's letter. And of course, like what always happens with new proteins, is those who do research with using mouse genetic models recognized this new finding, and they first developed a mouse which was knocked out, deficient in LEAP, primarily because the gene was adjacent to a transcription factor they were studying called USF2, upstream stimulatory factor 2. And as it turned out, these researchers who were studying USF2 then found that their mice developed severe tissue iron overload. And they went ahead and realized that just downstream of USF2 was this protein which had been called LEAP, and they renamed it hepcidin. And then a year later, when a transgenic mouse was developed in which the protein is overexpressed, you can see then that that mouse developed severe iron deficiency. So now it's known that hepcidin, its mRNA, is expressed in the liver, and it's an endocrine hormone, if you will, which has target tissues, including the duodenal enterocyte and the spleen, and it has effects on the liver as well. So that's the way why I call this the endocrine aspects of hemochromatosis, because the key regulator of iron absorption is truly an endocrine hormone, hepcidin. And the hepcidin is found in the blood, and this shows its mechanism and how it regulates ferroportin. So ferroportin is the target, if you will, for this endocrine hormone hepcidin, because the hepcidin binds to a complex containing ferroportin and results in the recruitment of a transcription factor known as JAK2, and they get phosphorylated, and this phosphorylated complex then becomes internalized in these pits, and they form endosomes, and then the hepcidin results in the degradation of the ferroportin through this mechanism. So when you have a great deal of hepcidin in the blood, you degrade ferroportin and you absorb less iron from the GI tract. And that's shown here. Lots of hepcidin going through the plasma into the duodenal enterocyte. The ferroportin then is degraded. It's a complex with hepcidin. There's less ferroportin, and there's only a small amount of iron absorption. Whereas if something disrupts the production of hepcidin, there's less hepcidin, and therefore more ferroportin, and therefore much more iron gets absorbed. So now the other part of an endocrine system is feedback regulation. And the feedback regulation in this situation is accomplished by the iron transferrin complex. So the iron then is circulating in the blood because that was what was absorbed by the ferroportin. And the iron transferrin important complex then regulates the gene hepcidin. And how exactly it does that is quite controversial. There's many papers that I read about this in the past year or two, all of whom have a slightly different slant on exactly what's going on here. But one simple mechanism is that the transferrin iron complex binds to the transferrin receptor type 1, a ubiquitous transferrin receptor, and that it recognizes that and then activates this complex, which includes a protein called HFE. And the transferrin receptor 1 HFE complex then stimulates some intracellular transcription factor family known as SMADs, and they get phosphorylated, and they then, once phosphorylated and as a dimer, they move into the nucleus, where together with other proteins, they stimulate hepcidin synthesis. Another series of experiments instead suggests that when the transferrin bound to iron activates the transferrin receptor, the HFE moves to the adjacent transferrin receptor type 2, and it's the transferrin type 2 HFE complex which instead initiates the SMAD signaling. Now, the HFE is a very, very key molecule in that even though it's hepcidin, which is the endocrine hormone, it is not mutations in hepcidin which cause most cases of hemochromatosis. There are some cases of hemochromatosis due to hepcidin, but the vast majority of the cases are due to mutations in the protein known as HFE. And a few cases are also found in which the mutation is in that transferrin receptor type 2, supporting the importance of the transferrin type 2 receptor in the normal physiology. Now, there are alternate mechanisms to regulate hepcidin as well. And one involves a protein called homojuvelin and a protein which is another one of the TGF-beta family members called BMP6. And the BMPs also activate the SMAD signaling. And thus you can see that the BMP together with the homojuvelin and other factors also converge in this same downstream signaling pathway. Another way to activate hepcidin is through cytokines. Inflammation, especially the IL-16, which does that. And hypoxia negatively regulates hepcidin in a way accounting for the increased iron absorption, which allows for more hemoglobin production in the conditions of hepcidin. So, as it turns out, some cases of hemochromatosis are due to mutations here, the majority here, a few here, and then there are a type of juvenile hemochromatosis, which is a much more severe and early onset condition in which the mutations have been associated with this protein, HJB. So when the iron transferrin complex circulates in the blood, it has physiological functions, and it does so by entering into the cell and then getting absorbed into the endosome. The transferrin receptor is recycled, and it goes on to be able to bind additional iron molecules. And once inside the endosome, the iron then can be partitioned to ferritin. The same ferritin we've talked about before is a storage form of iron. Or alternatively, it can function in the variety of physiological enzymes and proteins that I mentioned before. There's also a very beautiful molecular mechanism for regulating how iron regulates its own entry and storage in the cells. And this involves another protein which is called iron regulatory protein 1. And this protein binds to RNA molecules, mRNA molecules, and it binds to the 5' regulatory region of the ferritin gene, and it binds to the 3' region of the transferrin receptor. So when these types of proteins occupy the 3' region here, they stabilize the mRNA. So there's less degradation of the mRNA, and there's less protein synthesis. And here, the protein then blocks the translation of the ferritin. So in situations where there's little iron, where the body senses iron depletion, the IRBP1 is bound to these RNAs. You get more transferrin receptor because the mRNA is stabilized, and in the setting of iron depletion, iron enters the cell. On the other hand, you get less transferrin produced, and therefore the iron which enters the cell doesn't go into a storage pool, but instead it's used for cellular function. And when iron is present and can bind to the IRP1, the protein then dissociates from the mRNAs, and you get translation of ferritin. So that's the typical high level of ferritin which we associate with iron excess. And the cell protects itself by making less transferrin receptor, because in the absence of the stabilization, the mRNA is degraded and there's less transferrin receptor. So you can see that there's this beautiful, intricate homeostatic mechanism to control iron entry into the cell. So, while all organisms are dependent on iron for survival, and in state of iron excess, where the iron begins to exceed the transferrin binding capacity, you begin to form free radicals instead, such as oxygen free radicals and hydroxyl free radicals, and these damage cells. They damage the sarcoplasmic membranes, the lysosome, mitochondria, DNA structure of the cell, and ultimately cells die. So in summary then, the hepcidin is produced by the liver. It's controlled by a series of other genes and proteins. It circulates into the blood. It regulates the metabolism, or the degradation of ferroportin, which then allows more iron to be absorbed, and downregulates the amount when the hepcidin goes up. And when there's hepcidin deficiency, then there's more ferroportin on the duodenal enterocyte. You get more iron into cells, and that begins to damage the cells throughout the body, with an increase in the plasma iron pool because there's more iron absorbed from the circulation. So the clinical association of bronze diabetes and cirrhosis was first identified in the French literature in the late 19th century, these three pivotal papers. And we now know that hepatocytes are an important cell that's damaged in iron excess. The pancreatic beta cells and the skin, as in the initial papers. And the other cells which are damaged very frequently are the gonadotroph, causing hypogonadotropic hypogonadism, the cardiomyocyte, producing a cardiomyopathy, and interestingly, the synovium of the hands, involving the second and third MT joints, producing a type of arthritis in the hands, which is very, very interesting. So when any of those abnormalities are found, one thinks of identifying whether there's iron excess. And the way to identify whether there's iron excess is to measure the amount of ferritin, that protein that we discussed before, iron binding protein, and to look to see if the transferrin saturation is high. And I'm sure you're all familiar with this. But even though hepcidin would be the desirable candidate for assessing hemochromatosis, so far there are no approved assays for hepcidin. That people have been studying it in blood, in the urine, and they haven't been able to figure out how to measure it accurately and best understand how it relates to this physiology. So we're still measuring transferrin and ferritin. And here are a group of people who have hereditary hemochromatosis, and the dark bars are the men and the open bars are the women. And you can see that the majority of patients who are affected have elevated ferritin levels, but not all the patients. And that when you get to a very high level, like the level we saw in our patient, very few women have that very high level, and only about 40% of the men. Similarly, the transferrin saturation is elevated in about 80% of the men and about 70% of the women. And in general, adult women are thought to be protected from iron absorption because of the extra blood loss. But there's also a series of studies which show a connection between estrogen receptor signaling and many of the proteins which I described to you, implying that estrogens play a role in influencing iron absorption as well. So the problem with these biomarkers is that they're not entirely sensitive. Many of the patients, as I showed in the last slide, have normal values, and they're also not very specific. So in general, a high ferritin value is identified as a value of more than 200 micrograms per liter in women and 300 micrograms per liter in men. And in fact, the majority of people who have high values don't have hemochromatosis. They instead have other factors, other illnesses, which result in increased ferritin production. Most notably inflammation, various types of arthritis, will increase the ferritin. The metabolic syndrome and liver disease, especially alcoholic liver disease and viral hepatitis, EB virus hepatitis. Some people write that if the value is normal, it excludes iron overload. So in a way, the ferritin is a useful test as a diagnosis to exclude the disease, rather because there are so many false positives. The transferrin saturation may be a bit better, but it too has problems. Generally, it's defined as abnormal when the value is more than 45% in women and 55% in men. But there's a great deal of biological variation in circulating iron. There's a diurnal rhythm, and there's a meal-associated change, which results in impacts on these factors. In addition, the transferrin itself is suppressed with inflammation, and there are a wide variety of reference methods for TIBC, which confuse the field as well. And our patient, who I presented, had the metabolic syndrome and had transferrin suppression, and it seems much more likely that her disease was not hemochromatosis because of the normal hepatic iron and the genotype that we found, but instead that her iron parameters or iron excess was really due to other factors which influence iron transport and partitioning. So according to the European Association for the Study of Liver Disease, the hemochromatosis is divided into 3 stages. Stage 1, patients who have a genetic disorder but no increase in iron stores, they have a genetic susceptibility. Stage 2, people have increased iron overload, but they don't have any tissue damage. And stage 3 individuals also have tissue damage. So not everyone, and in fact, only a minority of people seem to progress from stage 1 to stage 3. So there must be other factors, alcoholism, which is one, the metabolic syndrome is another, and a wide variety of other genetic as well as environmental factors seem to influence how the genetic disease progresses to the clinical disease. And we can see that by just looking at the prevalence of hemochromatosis by various criteria. Back in the 1950s, sorry about that. Back in the 1950s, the prevalence was only 1 in 20,000 individuals based on the hospital admissions for cirrhosis, pigmentation, diabetes, and arthralgia. But on the other hand, if you do a genetic survey of the gene mutation, which I'll show you is the most common one, the C282Y mutation, you can see that 0.3% of the population is homozygous for this disease. That means that the carrier frequency is close to 1 in 10, and that means there's lots of us here in this room who are carriers for this gene. So back in the 1980s, get this right eventually. The symptoms and signs of hemochromatosis were those which were demonstrated by people who had the clinical disorder, because the genetics, of course, was not yet understood. And these are individuals who had a liver biopsy with having parenchymal iron, so no one would argue that they didn't have clinical hemochromatosis. And the symptoms that they would have was very, very common. They would have weakness and lethargy, abdominal pain, arthralgias, loss of libido and impotence, and congestive heart failure. And the physical and diagnostic findings would be cirrhosis, of course, with a large liver and splenomegaly. They look hypogonadal, with loss of body hair, and often with breast enlargement. They have small testes very frequently, and skin pigmentation and clinical diabetes. But on the other hand, now, if we take individuals who are identified as having hemochromatosis based on their genetic mutation, the majority of the people are asymptomatic. So this is 200 individuals who were 282 homozygous, so it's a cysteine to a tyrosine mutation, which is an important mutation. And about 200 people were found in 40,000 or so who were screen for the disease. And they divided them into those who had markedly elevated ferritin levels and mildly elevated ferritin levels. And these are just males and females. You can see that the most severely affected are usually males for the reasons I said before. But you can also see that the number of subjects who had symptoms is few. A very small percentage of the patients had fatigue, arthritis, and a history of liver disease. And they didn't comment on hypogonadism in this paper. So I've already alluded to the fact that the key mutation is in the HFE gene, and it occurs in this amino acid 282 with a mutation changing the cysteine to a tyrosine. It's a single base pair mutation. And this mutation occurs here at the base of the extracellular portion of the hepcidin protein, where it interacts with beta-2 microglobulin, which tends to stabilize the HFE. So when the mutation is present, the interaction with beta-2 microglobulin doesn't work well, and the HFE protein becomes readily degraded. It does not appear on the surface of the hepatocyte as it should, and therefore it's not able to activate hepcidin production. Two other common mutations are in mutation 63 and in 65, but these are in distant portions of the molecule, and these mutations, although common, much less frequently are associated with clinical disease. Sometimes you'll find a compound heterozygote, because this is a very common mutation, even more so than this mutation. And sometimes you'll find patients who have one allele having the 63 mutation and the other have the 282 mutation, and those people do get symptomatic. So hereditary hemochromatosis is primarily a disease of Caucasians. So this is a survey of the genotypes in people in Washington, D.C., Birmingham, Alabama, Irvine, California, Portland, Oregon, Honolulu, and London, Ontario. And you can see of 44,000 Caucasians who they genotyped, the disease was found in 0.44%. In Native Americans, it was 1/4 as common, 0.11%, even less so in Hispanics, and vanishingly unusual in African Americans. And that's true with the compound heterozygote mutations as well, and with H63D homozygote. So hemochromatosis is primarily a disease of European Caucasians. So I've mentioned already that the majority of patients have a mutation in HFE, and you have to have two affected alleles, so it's an autosomal recessive disease which disrupts hepcidin synthesis and results in arthropathy, hyperpigmentation, cirrhosis, diabetes, cardiomyopathy, and endocrine dysfunction. And this accounts for 80 to 85% of all of the people who have clinical hemochromatosis. There's another mutation in transferrin receptor type 2. I alluded to that earlier. And it produces a phenotype much like transferrin, much like the type 1. And this is called type 3 hemochromatosis. Type 2 hemochromatosis, 2A and 2B, are a juvenile form, which is much more severe and occurs before age 30, and is characterized by hypogonadism and cardiomyopathy. And the proteins involved are the HJV, the homojuvelin, which I showed you, one of the regulators of hepcidin synthesis, and the hepcidin gene itself, which is called HEM. These are all autosomal recessives. There is a disease which is autosomal dominant, and it's a constitutively activating form of ferroportin. It's quite rare. So this compares some of the clinical findings in the juvenile form with the adult forms, type 1 and type 3, the HFE and the transferrin receptor type 2 mutation. And of course, the major characteristic is a much younger time at which the patients present clinically. And although the transferrin saturation and serum iron aren't any different at the time of diagnosis, These individuals much more frequently have hypogonadism, much more frequently have cardiomyopathy, and much more frequently have glucose intolerance. So this is type 2 or juvenile form. And unlike the HFE mutations, the other forms of hemochromatosis are not characterized by any one specific unique mutation. A variety of mutations in each of these genes has been recorded in the literature. So to identify mutations for these genes, one needs to do sequencing, whereas one can use restrictive fragment polymorphism testing for the HFE, and I'll show you that in a minute. So we generally think, when we think of Mendelian genetics, we think of individuals who are who have an autosomal recessive disease, and they have two alleles which produce a non-functional protein, and they're deficient then. In this case, they'd be deficient in HFE. Whereas if you have only one abnormal allele and one wild-type allele, then you're going to have relatively normal function, and you're not going to have a clinical phenotype. And most of the carriers, like the 1 out of 10 of us in the room who carry either the HFE or the H63D, are pretty healthy, and we don't have any phenotype. But there's another type of genetic inheritance, which is now being called multigenic or digenic. And you can imagine that if there are all of these transcription factors are playing a role in the activation of hepcidin synthesis, like I showed early on, that if several of the transcription factors are only partly functional, when you put those mutations all together, you may wind up with a clinical phenotype. And that's been found in a variety of diseases. Cystic fibrosis, I was going to bring that up for our chair who's not here, and hypogonadotropic hypogonadism, a disease that I'm interested in, and also in hemochromatosis, as shown here. So here is an individual with a juvenile form of hemochromatosis. Okay, we'll get this right eventually. With a very high transferrin saturation and a very high ferritin. And he's found to have a mutation for the HMP gene, which codes for hepcidin, but he's a heterozygote. He's got one abnormal allele and one normal allele. But on further testing, he's also a heterozygote for HFE. So he's got one normal allele and one mutant allele. And many of his family members died prematurely, and really, well, really the only informative information here was his mother, who likewise has the mutation in HAMP, and she's again a heterozygote, but she has a wild-type HFE sequence. So the mother is not affected clinically. She has one of the two mutations that her son does. Her son presumably inherited the HFE gene from his father, but he only inherited one, and his father then could not be tested. So here's an individual who has a type of compound heterozygous for two genes, both of which activate hepcidin production. So I became interested in hemochromatosis a long time ago. I was telling Roberto that I saw this patient when I was a clinical research fellow at the NIH, and he was sent to the endocrine branch because he had hypogonadism. And he was about 24 years old. He'd been in the military. He'd never shaved at his beard ever, and when all the other guys shaved, the sergeant told him to go shave anyway, rather than saying, you know, maybe this guy needs a physical exam. And when he was sent to us, he, you know, looked much younger than his stated age, with no body hair. His testis size was reduced, and you can see he has a very dramatic skin pigmentation. Sadly, he also had atrial fibrillation, and a few months later he died of congestive heart failure. And This shows the secretion of luteinizing hormone into the blood in normal men, where blood samples are drawn every 10 minutes for 12 hours. And because of the activation of the pituitary by GnRH, produced by the hypothalamus, the pituitary releases LH, which circulates in the blood and stimulates the testis. And you can see that these bursts occur in somewhat of an irregular interval. And also there's also some amplitude variation. And I did lots of research like this in the way back when. And in contrast to the normal individuals, the patient with hereditary hemochromatosis, who is severely hypogonadal, had basically no spikes, no bursts of LH at all. And when he was treated with GnRH, which would initiate LH secretion in children, for example, it didn't do anything. It hardly increased his LH as well. So this suggested that his pituitary was somehow resistant to the GnRH. And now we can show that that's true because of iron deposition in the pituitary. So the MRI is now being used to identify how much iron is present in the liver and in the heart in people with hereditary hemochromatosis. And you can see how dramatic the decrease enhancement is in liver in this affected patient in comparison. Normally the liver looks like the spleen. Certainly it's very different here. So this is a sagittal view of the brain, and this area is where you find the pituitary gland, and it's expanded over here. This is the optic chiasm, the pituitary stalk, and the pituitary gland itself. And you can see that inside here is this dark area, not as dark as that, but it should all be homogeneous, and certainly it's not. And this shows you, just like the biochemical test did before, that the iron is found in the pituitary gland, and it's preventing the gonadotroph from responding to GnRH. And in fact, hypogonadotropic hypogonadism is a very, very common finding, and often the first finding in retrospect in adult men with hereditary hemochromatosis. Here's a group of patients, middle-aged men, published in 2005, with a very, very liberal low cutoff for testosterone. Normally we use a value of 8 or even 10 nanomolar, and their normal range started at 4, and they concluded that 25% of the men at that clinic were hypogonadal. You can see low testosterone levels together with low LH levels, which increased slightly or not at all in response to GnRH stimulation, indicating how frequently hypogonadotropic hypogonadism is found in hemochromatosis patients. And so that in adults, there are far, far more affected males than there are females, whereas in the juvenile form, males and females are affected. Similarly, as I said before, this may have to do with the protective effect of estrogens as women get older, or the lack of menstrual bleeding when the iron's accumulating in the younger individuals. Quite interestingly, even though the gonadotrophs are disrupted, the other pituitary cells are not. So here are the cortisol levels in the hemochromatosis patients in 2 trials. And the patients are given an injection of ACTH, a cortrosine test that we all use to assess ACTH and adrenal function. And if anything, the cortisol levels in the patients are a little bit higher than in the controls. And of course, they respond quite nicely to cortrosine. So there's nothing wrong with the adrenals or with the corticotrophs. Similarly, there's hardly anything wrong with the thyroid and the thyrotrophs. It took me a while to find a single case of secondary hypothyroidism in hereditary hemochromatosis. There is one, but just one. And here's an individual, again, with a low testosterone and low LH. And here the T4 and free T4 are presented in terms of SI units, nanomolar and picomolar, so you're not used to these numbers. But in comparison to the normal range, this person has a low total T4 and a low free T4 and a low TSH. So we know that that person has TSH deficiency. And you can see they even show the results after several years of phlebotomy that the situation improves. So Hypothyroidism has been reported, but it's quite rare. In pituitary diseases, the hormone which is disrupted most often is growth hormone. And the way we test for that is by producing hypoglycemia. And the brain and pituitary secrete growth hormone in response to the hypoglycemic stimulus. And a normal value is maybe 6 or 8. They use 10 as the cut point here. And you can see that the hemochromatosis patients, compared to the normal, there are a few more hemochromatosis patients below 10, but overall, the majority of the patients, as in the controls, produce a robust amount of growth hormone. So there's very little evidence for the somatotroph damage. The posterior pituitary is also rarely abnormal, and one case I found was a woman who developed diabetes insipidus in pregnancy. In the 3rd trimester of pregnancy, women tend to make lots of urine, in part because the placenta produces a hormone called vasopressinase, and the vasopressinase then metabolizes the vasopressin, and normal women make more vasopressin to compensate. But if there's something the matter with your posterior pituitary and you can't make more vasopressin, then you'll develop diabetes insipidus, and that's what happened to this lady. She was pregnant. She developed polyuria up to 12 liters. Well, 14 liters of urine. So, with a high plasma osmolality after she was dehydrated, and a low urine osmolality, and a low vasopressin level. And then when she was given vasopressin, her urinary osmolality rose dramatically, indicating that she could respond to the vasopressin. And these are the typical findings in central diabetes insipidus. And I present this because in this case they showed the RFLP, and I wanted to show you exactly how the mutation is identified when you might order it. So it's based on doing PCR and then treating the PCR product with an enzyme, a restriction enzyme. And if the mutation is present, the restriction enzyme recognizes the adjacent base pairs and cleaves the product into two fragments. When the wild type gene is there, the enzyme does not recognize the base pairs and no cleavage occurs. So here's the PCR product in a wild type individual. Here's this woman who doesn't have any 486 band at all, but has only the 2 smaller pieces. And here's a heterozygote who has both the 486 band and the smaller pieces. So this is how the test is done if you ordered it. And in fact, she had a very high ferritin level and a high level of iron and TIBC, and had hepatomegaly. All of this was unrecognized when she was pregnant. So diabetes mellitus is also an endocrine complication of hemochromatosis. Diabetes is usually divided into type 1 and type 2, and then a variety of other conditions known as secondary diabetes, and among those is hemochromatosis. And here we see a pancreas which is stained with Berlin blue, a stain which is used to identify hemosiderin, which is a protein which is a disrupted type of transferrin. So as iron bound to hemosiderin is no longer in a releasable pool, and instead the hemosiderin remains in the tissue, and you can see that it stains blue with this stain. So there's a lot of iron in this pancreas, including this islet. And here we see a normal individual in which immunohistochemistry was used to stain for insulin. And here we see the islet from a patient with hemochromatosis, in this case due to transfusion iron overload, which I'm not talking about, but I couldn't find one with hereditary hemochromatosis. So you can see that there's marked depletion of the insulin content, as well as the content of glucagon in comparison to the control. And here we have the results of glucose tolerance tests in 30 patients with hereditary hemochromatosis, and their controls with siblings and relatives who are in the open box. And here they've divided the patients into normal glucose tolerance, impaired glucose tolerance, and those with diabetes. And you can see that no one in the control group had diabetes, and that the chance of being impaired glucose tolerance was much higher in the patients than the controls, and many more controls were normal. And among the three groups of hemochromatosis patients with normal, impaired, and diabetes, you really don't have a statistically significant difference in serum ferritin or age. But one thing that fell out is that the people with diabetes tended to be much heavier than the hemochromatosis patients with impaired glucose tolerance. And that gets back to what I said earlier, and that is that many of the phenotypic abnormalities in this disease result from other issues which work in conjunction with the iron overload. And in this case, it's presumably the insulin resistance, which is associated with diabetes, which resulted in that those people being symptomatic. So when iron enters a cell, the textbook explanation for the damage has to do with the reduction of the free radicals and reactive oxygen species, and damage to mitochondrial membranes and myofilaments and other parts of the cell, in this case the cardiomyocyte. But really, why only certain cells get damaged or is not entirely clear. And it's very, very dramatic for the pituitary, where we see there's iron in the pituitary in the MRI. We see that the majority of the people have LH deficiency. But on the other hand, the TSH, the vasopressin, the ACTH, the growth hormone, they're okay. So why are some cells more sensitive than others? And I think that's a very important future area of investigation. And if you were to read the textbook, they would say that it's the cells that have more mitochondria or myofilaments, but I don't think that's true, at least in the pituitary. And a few years ago, a preliminary experiment together with John Eaton, which I hope to pursue in the future, when we took two cell lines, one a gonadotroph cell line known as alpha T3 cells, and then a corticotroph cell line known as ATT20 cells. And what John suggested is that we can assess the iron entry into these cells by loading them with a dye called calcine, and calcine is a fluorescent dye. And when iron enters a cell, it extinguishes the fluorescence. So here we incubated the cells overnight with increasing concentrations of iron, and you can see that the fluorescence was suppressed in a concentration-dependent manner in the gonadotrophs, but there was no effect on the fluorescence in the corticotrophs, implying that it was easier for the iron to enter this cell line than this one, or perhaps the iron was rapidly exported from this cell line and not this. So this seems like a worthwhile avenue of further investigation for us. So going to summarize now, this is from the American Association of Liver Disease, and what they would do is if you identify people who are either symptomatic or asymptomatic and have clinical findings suggesting hemochromatosis, or you have a known first-degree relative of a hemochromatosis patient, then you should, of course, evaluate them. And in these individuals, you don't begin with the genotype, it's too expensive, but instead you measure the serum ferritin and transferrin saturation. And if either or both are elevated, then you do the genotyping, which is primarily for that HFE283, as I showed before. And that should certainly be done in all first-degree relatives. And if the homozygous mutation is found, then the individuals get therapeutic phlebotomy. They suggest that if the ferritin level is more than 1,000 or liver enzymes are elevated, that the individual should have liver biopsy. And because so many of the other mutations don't in of themselves cause the clinical disease hemochromatosis, they suggest that if you do find some of the other mutations, as in our case where we found the H63D mutation, they suggest that you look around for other types of disease which might be contributing to the clinical picture. So the treatment is still with phlebotomy, even though you'd think that 10 years or so after hepcidin had been developed, that pharmaceutical companies would quickly develop a hepcidin analog or something which stimulates hepcidin secretion, or perhaps something which binds to or blocks ferroportin absorption of iron. All of those are logical, novel therapies of hemochromatosis. And yes, they're all in development, but in a quick clinicaltrials.gov search yesterday, still there are no clinical trials of any of these novel molecules out there. And we're still left with phlebotomy, which removes the iron, but of course doesn't cure the people of the increased iron absorption. And the way that the iron is removed is about a unit every week or so, and you want to try to produce mild iron deficient state without producing anemia, because the anemia then will increase the iron absorption. And you follow the ferritin level, and you stop the ferritin, stop the phlebotomies when the ferritin reaches 50 to 100. Remember, our patient, the value was more than 1,000. And it usually takes more than a year. So weekly phlebotomies for more than a year to get rid of all that iron. And then when the patients go into maintenance, you phlebotomize them 2 or 4 times a year to maintain the serum iron at this level. And they recommend that you avoid vitamin D supplements. It appears that vitamin C somehow seems to exacerbate some of the complications, although I couldn't find very good documentation. So here you can see that it works. These are men with low testosterone who were phlebotomized for quite a long time. And you can see that there is an increase in testosterone, although the increase is variable. And this seems to be the case for all the endpoints, the cardiac endpoints, the liver endpoints. Some people seem to get better, and some people don't. So in summary then, hereditary hemochromatosis is the most common autosomal recessive disorder in Northern Europeans, affecting 1 in 200 persons. The common downstream mechanism of iron overload is abnormal regulation of the hepcidin ferroportin axis, leading to failure to prevent excess iron from entering the circulation. Most hereditary hemochromatosis patients have a missense mutation in the HFE gene at locus 282. Heterozygotes are not iron overloaded. An enhanced intestinal absorption of iron leads to multiple organ damage in the liver, in the pancreatic beta cell, in the joints, in cardiomyocytes, in hypogonadism, and in the skin changes. And that you need a high index of clinical suspicion to diagnose early disease. That selective hypogonadotropic hypogonadism is a common early clinical finding. And finally, phlebotomy remains the treatment of choice. So thanks very much for your attention. Thank you. So if there are any questions, please. Or comments? Comments about the skin? None?
You mentioned there are some approaches about science trying to maybe synthesize analogs. How close are we to some cleaner hepcidin analogs?
No, I haven't found any company which has yet begun a clinical trial. All of the analogs are being studied in vitro and in animal models. So the problem also is that none of the animal models are really that good with respect to the organ damage. It takes a while. And I guess showing that the drug is efficacious in an animal model has also been a little difficult.
So, I have a couple questions. The first is, what's the usual mechanism of entry of iron into the cell? And does that differ between the cell types? So are the ones that are more susceptible, does iron enter more easily?
Well, that's what I tried to show with the gonadotropes. The normal entry is through the transferrin receptor complex. So transferrin bound iron is the physiological mechanism where iron affects all the cells to produce all of those chain enzymes and proteins that I showed in the first slide. So when iron enters through that transferrin receptor, the receptor gets internalized, and the iron gets processed and partitioned to ferritin, and the cell is protected from the iron by binding to ferritin. When the transferrin saturation is exceeded and you get free iron into the blood, then iron seems to enter into certain cells selectively, the cardiomyocyte, the gonadotroph, the pancreatic beta cell. And like I said, the general textbook knowledge is that those cells are more sensitive, not because they get more iron in them, but because their organelles are more sensitive. And I don't think that's really applicable, at least in the pituitary. And then we did this preliminary experiment which suggested that free iron may enter a gonadotroph, admittedly a tumor gonadotroph, a cell line, more readily than the ACTH-producing cell. So all cells have the transferrin receptor type 1, and that's how they physiologically process iron for all their biological function.
Okay, and then the second question was, you said the iron absorption is still under hypoxic control even with this mutation. So you said you don't phlebotomize them to anemia.
Right.
Because this will stimulate more iron absorption.
Right.
So there is still some regulation
Correct.
of iron absorption even in this disorder.
Correct. Right. So the signaling pathway controlling the hepcidin is sort of downstream from the control. There's nothing wrong with hepcidin in the HFE mutation patients. The hepcidin is just an innocent bystander, if you will. It's not activated because it's not being told to in a way that you can get, downstream endocrine dysfunction because of the pituitary dysfunction. So if you activate the hepcidin in another way, then you'll get a lot of hepcidin. That's why I said one of the other potential treatments would be to come up with a drug which stimulates hepcidin and sort of circumvents the deficiency in HFE. Well, thanks for your attention.